So good morning everyone, it's really nice to have you here in the early morning, and I hope in the next hour or so you will learn a lot and understand a little bit more about our MaxArt Annexin 5 portfolio. So as you can already see, we're going to basically talk about how you can find the fittest sperm today, and in that way how you can improve the selection of sperm cells for the IVF process in your lab if you're working in an IVF lab. Very shortly at the beginning, I would like to introduce you to our agenda. We'll start with a small introduction and then proceed to a more biological background on how the effect of apoptosis and DNA fragmentation have effect on the IVF outcomes. And we're going to talk about how the MaxArt Annexin 5 system works and how it can afterwards improve the quality of sperm preparations. And finally, we're going to look at some data on how did the MaxArt Annexin 5 system perform both in the lab and in the clinic. So for those of you who are new to Miltenyi Biotec, we are quite an international company with already 30 years of experience in areas like immunology, stem cells, neuroscience, cardiovascular research, cancer as well. And one of our newest clinical applications is, of course, the assisted reproductive technology portfolio, which is represented with MaxArt Annexin 5. Now, why is this product essentially important to us, and why do we really think that it will benefit mostly the people who are working in the IVF lab? So, probably you have already been introduced in the media to the male fertility crisis, which was represented by a couple of headlines. For example, like, we're heading for a male fertility crisis and we're not prepared, or 6 things that are harming your sperm count. A male infertility crisis is coming, the Middle East can help. And the male infertility crisis in US has experts baffled. Some even were skeptical if there is a male fertility crisis at all. Now, all of these media attention came after one study in Israel, which pointed out that the average sperm count has dived by 52% in the past 4 decades, and that men are leaving it later and later before trying to have a child. And this, of course, has both scientists and clinicians baffled, and they don't know why this count is falling or even whether the decline is reducing fertility, although the logic suggests that it is. And the fact is that our understanding of key male fertility questions has moved very little from the 1990s. We still don't know what causes most cases of male infertility, even though we know how this represents itself clinically, which means we don't have the tools to correct them, or we don't know how to proceed with these patients, to say the least. Another point is that historically, most of the fertility both treatments and research has been really pointed towards the female part of the story, which is why we have much more tools to deal with the female part of the fertility story, unlike the male part. Which is why we at Miltenyi Biotec believe there is a huge space for improvement, especially in the male fertility field. Now, this brings me to the second point, where I want to discuss with you the effects of apoptosis and DNA fragmentation in spermatozoa, and how do they have influence on our IVF outcomes. So within a sperm cell, what we have here are 4 major apoptotic changes, and they display themselves first as a plasma membrane changes, which are represented by the externalization of the phosphatidylserine from the inner leaflet of the sperm plasma membrane to its outer surface. This proceeds, of course, with cytoplasmic changes, which are represented by caspase activation, together joined by mitochondrial changes, where the mitochondrial membrane potential is disrupted. And finally, we have nuclear changes represented through DNA fragmentation, which can later on be also visualized in how the sperm looks, so to speak, or its morphology. And these 4 apoptotic biomarkers, for example, the externalized phosphatidylserine together with DNA damage and aneuploidy, which is the abnormal chromosomal number in human sperm cells, contribute or are linked with failure of both in vivo and in vitro fertilization. They're most likely to impair the chances of fertilization and pregnancy, and are definitely detected in significantly higher levels in infertile men and those with varicocele, for example. And last but not least, are partially responsible for low blastocyst development rates, pregnancy rates, and recurrent pregnancy loss. Another thing is that this apoptotic or early apoptotic spermatozoa cannot be identified by morphology. And even though we have tools to really magnify the sperm cells nowadays in order to see which cell is morphologically normal or which has large nuclear vacuoles or small nuclear vacuoles, we cannot say for sure if those which are morphologically normal are not undergoing an early apoptotic process. Therefore, we know that apoptotic or early apoptotic sperm cells are morphologically indistinguishable from viable cells and are present definitely in all sperm samples to different extent. And we also know that current methods, for example, for sperm preparation are non-specific for early apoptotic sperm cells, such as, for example, the density gradient centrifugation, where we purify morphologically normal sperm cells and motile sperm cells. And we also have the swim-up as a second most commonly used sperm selection method of motile sperm cells. And here, for example, we only select for the motile sperm cells, but we do not answer if these motile sperm cells do not undergo early apoptotic sperm cells. And another sperm preparation, or rather selection, of course, technique is the pixie dish, and this is a step above the DGC and swim up in the sense that it is already incorporated to the ICSI procedure. And here we have selection of mature sperm cells based on their ability to bind to hyaluronan. However, none of these 3 or existing techniques removes apoptotic spermatozoa specifically, which is why there is a need of new techniques to target especially these apoptotic sperm cells. So, as I already mentioned, in a living sperm cell or in the intracellular membrane, we have the phosphatidylserine located in the cytosolic side of the plasma membrane. And during this early apoptosis stage, the phosphatidylserine is then redistributed from the intracellular to the extracellular site. And one crucial component of the MaxArt Annexin 5 system is the MaxArt Annexin 5 reagent, which binds to the phosphatidylserine on the outside of apoptotic cells, and in this way marks the sperm cells' further processes. And in such a way, the reagent, or the MaxArt Annexin 5 reagent, is used for the depletion of these apoptotic sperm cells with externalized phosphatidylserine. So the next thing that we have is that this general MAX technology is actually a gold standard for cell separation, and by labeling the phosphatidylserine positive apoptotic sperm cells with the Annexin 5 reagent, we can then proceed to the separation step and apply the mixture of reagent and sperm cells into a column which is attached to a magnetic field. And since the reagent is also, or the MaxArt Annexin 5 reagent is coupled to magnetic nanobeads as well, the labeled sperm cells will remain inside the column, inside the magnetic field. Actually, they will be retained while the viable cells are collected in the flow-through and can afterwards be used for subsequent IVF applications. Now, speaking about the Max microbeads, it's important to mention that they're very small and non-toxic, and they have a low level of cross-linking and no physiological activation. And There is no detachment required, and they're always found in suspension. They're only about 15 nanometers in diameter and composed of iron oxide and dextran, and are non-toxic and biodegradable. And very important to mention is that by labeling the cells, there's with these microbeads, their cell function is also preserved. And currently, they are the only magnetic beads which are used in clinical cellular treatments. So this is how also the Annexin 5 microbeads actually specifically bind and remain on the apoptotic sperm cells. Now, the product information which I want to share with you is that the Annexin 5, the MaxArt Annexin 5 system here is composed of 4 parts, and all these components are CE marked. So we have the reagent, which is used, as I said, to label the phosphatidylserine positive cells. We have the buffer, the binding buffer, which actually helps the binding reaction between the reagent and the sperm cells. We have the separation unit, and we have the CE marked columns. Now, aside from the separation unit, all other components are single-use components, which actually makes sense because, of course, you are working in the IVF lab and you want to really prevent sample mix-up. It's also important to understand the intended use of the MaxArt Annexin 5, which in this case would be the in vitro depletion of apoptotic human spermatozoa by depleting phosphatidylserine positive cells from fresh or cryopreserved or otherwise manipulated semen sample. Each component comes with a batch-specific certificate of analysis and offers greater flexibility because you actually are able to split the kit in the sense that the reagent is also offered in 1 or 6 bottles. So you can have it for 1 or 6 separations, and it has maximum shelf life of 5 months. While the buffer and the columns are offered for 6 separations, and they have a maximum shelf life of 3 years. And very importantly here is to mention that the entire system is tested for embryo toxicity using the mouse embryo assay, which really gives a sense of safety for working in the IVF lab, because the mouse embryo assay is the golden standard to test for embryo toxicity and even gamete toxicity compounds. Now we go to the 4th part, and here I would like to let you know about how does the MaxArt Annexin 5 actually improve the quality of sperm preparations. What we know is that the cells separated by MaxArt Annexin 5 can be actually used for all IVF applications, starting from conventional IVF, mostly for ICSI, and even for intrauterine insemination. And based on studies which were published, the potential benefits here would be better fertilization and pregnancy rates, lower DNA fragmentation index in the sperm cells, in the purified sperm cells, and improved semen parameters such as motility and morphology. And the MaxArt Annexin 5 depletion can actually be really easily incorporated in a daily routine in the IVF lab, and can be used either before or after density gradient or swim up. So if you first do, for example, density gradient or swim up and then proceed to the MaxArt Annexin 5 separation, this protocol will require an additional centrifugation step with a sperm washing medium after the separation in order to remove the binding buffer. But if you first do the MaxArt Annexin 5 separation and then you do the density gradient or the swim up, this protocol will save you 1 washing centrifugation step after separation. There is a certain flexibility of how you want to proceed and how you want to combine the MaxArt Annexin 5, and this is definitely dependent on how your laboratory is set up and on the experience of the personnel working in the lab. So here I would like to shortly introduce, for example, how a certain protocol would look like if you decide to first go with density gradient centrifugation and then perform the MaxArt Annexin 5. It takes around 25 minutes, and what you need to do after density gradient centrifugation is first to count the cells. So the minimum threshold here would be overall 100,000 to maximum 50 million total sperm cells. Please bear in mind that the total number of the sperm cells differs from the starting concentration of the sample. And after that, you can wash the cells with the binding buffer and centrifuge 5 minutes at 300 G at room temperature to remove the supernatant. Afterwards, you can proceed to labeling the sperm cells by adding the complete vial of Annexin 5 reagent and filling it up to a final volume of 500 microliters with the binding buffer. You proceed to basically the core step of the procedure, which is the incubation, which happens at room temperature for 5 minutes. And during this incubation step, actually, the reagent, the Maxart Annexin 5 reagent, is bound to the sperm cells, to the apoptotic sperm cells. During this time, you can already prepare for the next step by washing or inserting the column in the separation unit and washing it by binding buffer, by approximately using, for example, 1 milliliter. And this will, in a way, also activate the flow through in the column. After the 15 minutes of incubation, you can definitely apply the mix to the column and collect the flow through. The flow through, of course, will contain the Annexin 5 negative fraction, which represents, in this case, the viable cells. And the apoptotic spermatozoa, which are the Annexin 5 positive cells, will be retained in the column. Here you have an optional step to add another washing step with the binding buffer by adding approximately 500 microliters. And this step is simply optional because in the case you start with a higher number of total sperm cells, you would like to elute more. But if you are afraid of diluting the sample, for example, this step can be optional and you can definitely skip it. Then you would proceed to the last centrifugation step, which is again performed at 300 G for 5 minutes at room temperature, and you can remove the supernatant. And finally, you can resuspend the supernatant into a sperm washing medium, a sperm buffer, and keep it at 15 minutes at room temperature to 20 minutes at room temperature, and then proceed to any IVF treatment that you would be performing afterwards. So now I want to proceed to the fifth part, and that is how does the Maxart Annexin 5 perform both in the lab, in the clinic. And here I would like to first show you some flow cytometry data where we have measured the viability or the level of apoptotic sperm cells in samples prepared only with density gradient centrifugation and in samples performed by density gradient centrifugation and the Annexin 5 microbeads. So in the first case, where the sperm samples were treated only with density gradient centrifugation, we see that we have 70% of viable cells, while in the second case, where these two methods were combined, we have more than 94% of viable cells. So flow cytometry basically shows that the use of the Annexin 5 microbeads increases the percentage of viable cells. Another data that we get from the lab and from studies is that the use of the Annexin 5 microbeads yielded in sperm populations with higher normal morphology, higher mitochondrial membrane potential integrity, unchanged or higher sperm motility, of course, reduced DNA fragmentation and reduced caspase 3 activation, which is an apoptotic assay. Also, what we can see is that the percentage of phosphatidylserine positive spermatozoa is actually lowest in the case of density gradient centrifugation and MAX, while it is highest in raw sperm samples and slightly higher in samples performed with density gradient centrifugation. And here in this case, the loss of sperm during separation is not more than 1.2%, which also speaks about the specificity of the product. And the mean percentage motility before separation in a given sample was around 65%, and after separation, the motility of the EPS negative fraction was 76%, and in the positive fraction was around 41%, which also shows that the use of the Annexin 5 increases the motility as well. So this corresponds to enhanced performance data that we have done. So we have measured a couple of parameters in both fractions, in the phosphatidylserine negative and positive fractions, such as depletion efficiency. We did caspase activity assays, mitochondria membrane potential, and DNA fragmentation assays. So you can see that the depletion efficiency here in the EPS negative fraction is around 23%, while the caspase activity in the negative fraction is 13% compared to the fraction in the positive, compared to the activity in the positive cells, which is 55%, which shows that we have lower apoptotic levels in the negative fraction. We also have the same or similar data when we measured the mitochondrial membrane potential, meaning the EPS negative fraction had around 32% of mitochondrial membrane potential, and the positive cells had around 70, almost 70. And the most promising result here is, of course, the DNA fragmentation assay, which decreased by almost 20% in the APS negative fraction, down to 13%. So these are data which are coming from the lab, and now I will show you some data which are coming from the clinic and public. This is publicly available and published data. So this study, which was performed by Dr. Dirijan in Turkey, included 196 couples which were treated by ICSI, and he divided the group in he divided the patients in two groups, namely the study group, which involved the density gradient, centrifugation, and the Annexin 5 microbeads, and then the control group. And they measured basically three parameters, such as the cleavage rates, chemical pregnancy rates, and clinical pregnancy rates. In this case, the cleavage rate was around 10% higher. The chemical pregnancy were about 15% higher, and the clinical pregnancy rates were also around 12% higher in the study group. Another study that we have is from Professor Sheikhi in Egypt, and he also had a similar study setup, in the sense that he had a study group in the study group he had patients where the sperm samples were treated with density gradient, centrifugation, and Annexin 5 microbeads, and the control group was treated only with density gradient centrifugation, and this again was an ICSI study, which is very important to mention. So in the study group, he observed 12% higher fertilization rates, almost 10% higher cleavage rates, almost 7 to 8% higher pregnancy rates, and very interestingly, 13% of higher birth rate, which is one of the few studies which are examining the birth rates as well with this system. Another study, which is a meta-analysis, which had a look at 4 studies which worked with the Maxart Annexin 5. So one of them is also the study of Dr. Dirijan. This was also included in this meta-analysis. They had a look at 499 potential patients in total, out of which 367 reported pregnancy. And what they showed is that in the study group, which again was the density gradient centrifugation together with the Maxart Annexin 5, versus the control group, which was the density gradient centrifugation only, there is an increase of almost 15 to 16% of pregnancy rates. And finally, they concluded in their paper that MAX appears to be a safe and efficient method to select functional sperm with consistently good results. This technique may improve pregnancy rates when used to complement standard sperm selection methods in art. So another study here showed up. This is a study performed in 29 normal zoospermic samples. So what they did was they had a setup of washed sperm cells and density gradient sperm cells, and in both of these groups they performed a max. So they combined max with the washed and sperm cells and the density gradient centrifugation sperm cells. And they measured the sperm DNA fragmentation index and the protamine deficiency rate. Now, in the case of DNA fragmentation index, they saw that the highest decrease of DNA fragmentation index occurs in the combination of density gradient and max. So, as you can see here, in the fraction of non-apoptotic sperm cells versus the fraction of apoptotic sperm cells, so that's the orange graph, the decrease of DNA fragmentation is higher. They also use the protamine deficiency rates, and as you might know, protamines are small arginine-rich nuclear proteins that replace histones late in the haploid phase of spermatogenesis and are believed essential for sperm head condensation and DNA stabilization. So they compared also the protamine deficiency rates between non-apoptotic and apoptotic sperm in washed semen samples and those that underwent density gradient centrifugation. And they also showed that the protamine deficiency rates of non-apoptotic sperm cells are much lower when combined with density gradient and max compared to the apoptotic sperm cells treated with density gradient centrifugation and max. So the conclusion of this study was basically that the combination of density gradient centrifugation and max may be an effective method to isolate high-quality sperm with progressive motility, non-apoptosis, high DNA integrity, and low protamine deficiency in clinical use. Finally, this is also a study which is coming from Spain, and what they have actually shown here is that they took they made 17 comparisons of sperm samples with certain aneuploidies, and they also showed that the Maxart Annexin 5 system, or the columns, actually remove sperms with numerical chromosomal abnormalities and aneuploidies. And these data here actually represent the frequencies of the different chromosomal abnormality in the max eluded fraction and the fraction retained in the max column. And basically in all of these 17 comparison, with the single exception of nullisomy 18, the frequency of numerical chromosomal abnormalities was higher in the retained fraction, which again shows that the Maxart Annexin 5 is actually able to retain sperm cells with numerical chromosomal abnormalities. And finally, due to time, I would like to also show you some case studies or papers which actually show and prove the safety of the Maxart Annexin 5, and these are mostly cases of postnatal observations, such as, for example, the paper from Romani, where they showed that obstetric and perinatal outcome of babies born from sperm selected by max are pretty normal. And we have cases like single and twin ongoing pregnancy in 2 cases of previous ART failure after ICSI, performed with sperm sorted using the Annexin 5 microbeads. We have cases where a healthy baby was born after max, and again, another interesting case report where they used the Annexin 5 to cell sort cryopreserved spermatozoa from a male cancer survivor, which resulted in healthy twin newborns after 2 previous ICSI failures. So the references here are already given, and you're more than welcome to have a look at them. So this actually brings me to the summary of this presentation and webinar. We already showed at the beginning that the apoptotic spermatozoa decrease the IVF fertilization and pregnancy rates, that the existing techniques such as density gradient, swim up, or even PIXI are not very specific for apoptotic sperm cells, which is why we need new methods, such as, for example, the Annexin 5 system to remove apoptotic sperm cells. And the procedure, as I already mentioned, takes around 25 minutes and should be combined with density gradient centrifugation or swim up, because it is only then that the effect is fully seen. Please bear in mind that the Annexin 5 is very specific for early apoptotic sperm cells, but density gradient centrifugation and swim up still add a certain value to the overall sperm selection process, in the sense that they might also remove abnormal sperm cells which are not of apoptotic origin. So it's very important that the MaxArt is combined with these two methods in order to have the most optimal performance at the end. We've been through some of the scientific publication and case studies which show that the use of Annexin 5 microbeads in combination with other techniques, such as the density gradient, for example, may increase the chances of fertilization, pregnancy, birth rates, and may decrease DNA fragmentation levels, and that it is already safe for use in ICSI patients, shown by some of the case studies I presented at the end. So with this, I would like to thank you for your attention.